What do you see now? Do you see my PowerPoint? You do see it? It's a okay. Let me see if I can record this. Let's see if this will work. My record will work. I'm looking for the record thing. There it is. I think that's it.
Recording in progress.
Okay. I believe we're recording now. Okay. And you do see the full PowerPoint, is that correct?
Yes.
I hope.
Yes, not on presentation mode though.
Yes. Okay. Here we go. Slideshow from beginning. You see presentation mode now with one PowerPoint
Yes.
screen? Okay. All right. All right. I guess we're ready to go then. Here, let me get my tea here so I could talk. Okay, so today we're going to start with the heart. The heart consists of three layers, just like the blood vessels consist of three layers. Pericardium, myocardium, endocardium. And this is a summary of all those layers in this drawing. The pericardium actually is a bilayered sac into which the heart fits. There's an outer layer called the parietal pericardium and an inner layer called the visceral pericardium. The two layers of pericardium are separated by a space called the pericardial space. The flat, simple epithelial cells which line the pericardial sac are sometimes called mesothelial cells. Mesothelial cells are not unique for the pericardium. They also see in the pleura and in the peritoneum. All of those membranes are serous membranes because the mesothelial cells secrete a serous fluid, a watery proteinaceous fluid. So one of the principal purposes of pericardium, notably the mesothelial cells lining the inner surfaces of the pericardial cavity, is to secrete a lubricating fluid so that when the heart beats, friction does not interfere with the heartbeat as there's motion between the visceral and parietal layers of the pericardial sac during the beating of the heart. The myocardium consists essentially of cardiac muscle, and the endocardium, which faces the blood-filled cavities of the heart, the atrial cavities and the ventricular cavities, is also lined by a flattened epithelium, a simple type of flattened epithelium, a squamous epithelium, if you will. Often that's simply called endothelium, the specialized epithelium lining the blood-filled cavities of the body, including those of the heart, is an endothelium. The mesothelial cells of the pericardial sac and the endothelial cells of the heart that face the blood lie, since they're epithelial cells, they lie upon a basal lamina, and that basal lamina lies upon at least a thin layer of connective tissue. So just like other epithelia of the body, wherever you find epithelia, generally speaking, the epithelium will lie upon a basal lamina, which will lie upon a supporting connective tissue. Okay, there's some other details in this image here, which you could look at on your own. You've seen this image before, but it does nicely summarize a number of things that we're going to be talking about today. Here's a cross-section of the heart, and just by looking at this quickly, I can see three layers of the heart. By the way, all the images I'm showing you today have been presented within our classroom materials, mostly within the lab. A few of them come from elsewhere. So these are repetitions of material. I'm not giving you new material particularly. There may be some minor editing, but all these things are already available pretty much in the materials you've already received. I've simply reordered them a bit or edited them slightly to make them work for a brief review. So looking at this picture here, do you see my arrow, by the way?
Yes.
Okay, good. Okay, so looking at this, it's easy for me to find the epicardium because usually in the epicardium we can easily find adipose tissue. The surface membrane of the heart is a serous membrane containing adipose tissue in the underlying support for the mesothelial cells. There's connective tissue, as I said. Well, that connective tissue also includes adipocytes. So it's very easy to find adipose tissue in the membrane on the surface of the heart. The visceral pericardium, which is also called the epicardium. Now, this specimen of heart has been removed from the pericardial sac, so we no longer have a parietal layer of pericardium to look at in this picture. We really no longer even have an obvious pericardial cavity to look at in this picture, but I easily see the visceral layer of pericardium, which we also call epicardium. Often we'll simply call it epicardium. So here's that fatty layer, which is the epicardium. On the surface, there will be mesothelial cells, which secrete that lubricating fluid again. Of course, there will be some areas where there's less fat and more collagenous tissue in the epicardium, but in this picture, the fat is quite prominent. There are blood vessels that travel over the surface of the heart before they send branches entering into the heart. So you're going to find coronary arteries and a variety of venous channels as well. Here we see a fairly large vascular channel, which is labeled coronary sinus. This is a vein, not an artery. There's a muscle layer here, which is part of the myocardium. A thin muscle layer is the atrial myocardium. A thicker muscle layer here is the ventricular myocardium. And you see they seem to meet at this point right here. Now, I don't really see it well, but I presume there's some type of um, connective tissue insulation separating the atrial myocardium from the ventricular myocardium. In other words, part of the cardiac skeleton must be there to electronically isolate the atrial myocardium from the ventricular myocardium, must be sitting at that point, even though it's hard to see. Now, the inner layer of the heart is quite thin. Here's a little box showing you part of the inner layer. And this quite thin inner layer is called endocardium. The epithelial cells lining that endocardial surface, therefore, are the endothelial cells. We don't really see the individual endothelial cells in this image here, but we know they are there, just like we know the mesothelial cells are covering the epicardium. There's a specialization of the endocardial layer here as well. This specialization is a flap of tissue, which is a heart valve. The heart valve is really a bit of redundant endocardium, if you want to call it that. Some of the endothelial cells of the endocardium, the uh, cover the surface of the valve on both sides. There's an atrial surface here, and there's a ventricular surface to this particular mitral valve, and that those surfaces are covered by endothelial cells. And the core of the valve consists essentially of connective tissue, which is in continuity with the connective tissue which is part of the endocardial layer. Again, the endocardial layer consists of endothelium, a subendothelial basal lamina, and beneath that, there's some connective tissue. So the heart valve really is an elaboration or specialization arising from the endocardial layer of the heart. Let's talk a little bit more about pericardium, the outer layer of the heart. We're going from the outside in. This heart has been removed from its sac. Originally, the pericardial sac is a bilayered sac with a parietal pericardium, and the inner layer is visceral pericardium, which we often, again, will call epicardium. Well, what we do see here is the epicardium, which is fairly rich in fat. As you can see here, there's lots of yellow adipose tissue covering the surface of the heart. That fat is within the epicardium. Now, not all of the epicardium has adipose tissue. There are areas without much fat at all. For example, where I'm putting my pointer right now, there's not much fat, and over here there's some areas where there's not much fat. So depending where you took a section, you may or may not see fat, but most of the time it's quite easy to see fat, which has a cushioning influence on the surface of the heart. If you've got a blow to the chest, for example, the fat would cushion the heart and protect, by the way, the blood vessels, which are circumferentially traveling over the surface of the heart, the coronary vasculature, the coronary arteries, and also the veins of the heart. Here's a cross section through the epicardium of the heart with some underlying myocardium as well. The epicardium has a very surface here, which has a layer of flattened epithelial cells, which are difficult to see in this particular image, but I presume they are there to some extent and were there during life. During tissue processing, it's quite easy for some of the cells to fall off for various reasons, partly from handling, partly because there's some autolysis of tissue when tissue is removed from the body or the person expires. So there presumably would have been a layer of mesothelial cells here, and I do see some evidence of it, but it is not the easiest thing to see in this particular picture. Underneath that, I see some connective tissue right here, which is fairly collagenous, and then deeper, I see lots of adipocytes. I see some blood vessels. Here's a blood vessel right here, which with a nice muscular layer, so I think it's probably, or muscular media, I should really say, which is, I presume, a branch of a coronary artery. And then here's another side branch, seemingly that would have come off this vessel or a nearby vessel that now is penetrating into the myocardium of the heart. So this looks like a branch of the coronary artery system. So all these things together are part of the epicardium. And of course, we're not seeing that here. We probably would find some nerve twigs or even ganglia in this area if we were to look around enough. And this is just myocardium. We can't see that much detail at this magnification, but our goal here is to really get a broad sense of the layers of the heart. So we don't have to see a lot of detail to conclude that we're looking at epicardium with some myocardium beneath it. Here's another picture showing you, again, epicardium. It's easy to identify this as epicardium because there's lots of adipose tissue within it. On the very surface, which I presume is facing the pericardial cavity, I see a simple epithelium here, and this simple epithelium is labeled M for mesothelial cells. So these cells would be secreting a lubricating serous fluid into the pericardial cavity. In the deep layer of this tissue, I see some muscle cells, so I presume these are muscle cells of the myocardium. And that's pretty much all I want to say about that. Again, just to reiterate, epicardium is also called visceral pericardium. Can things go wrong with this surface membrane? Sure, they can go wrong. One problem that could go wrong is when excess fluid accumulates within the pericardial cavity. For example, if someone had a blow to the chest, it may cause rupture of blood vessels and bleeding into the pericardial cavity, and they get what you might call a hemopericardium. Um, if someone had inflammation in the pericardial cavity because of infection, for example, a virus infection, fluid may accumulate. In that case, it would be inflammatory fluid, not blood per se. This particular picture shows more of a blood clot. Blood had entered into the pericardial cavity and then clotted in situ. So the blood clot has actually formed a cast of the pericardial cavity. Now, when fluid accumulates in the pericardial cavity, that's called an effusion. If a fluid accumulates to an extent where the person is in real health trouble, that's called a cardiac tamponade. If there's sufficient fluid in the pericardial cavity, it, for example, would make it very difficult for the heart to expand during diastole and fill up during for the heart chambers to fill up during diastole. If they cannot fill up during diastole, they cannot pump sufficient blood during systole, and this person would have heart failure. And this particular picture, by the way, it looks like somebody's taken a scissors and slid open the pericardial sac. In other words, they've taken a scissors and slid open the parietal pericardium, exposing the underlying blood clot within the pericardial cavity And again, this is an example of a cardiac tamponade. Sometimes because of inflammation or because of tumor metastasizing to the pericardium or because of old hemorrhage or old surgery, there may be some scarring or fibrosis of the pericardium. The pericardium gets thickened, and the layers of visceral and parietal pericardium may even get adherent or stuck to each other, like you see here, so that the pericardial space becomes obliterated. You're seeing that here in this particular image. The pericardium overall is thickened. The pericardial space has been obliterated because of adhesion between visceral and parietal layers. And the color of this, by the way, is a kind of a grayish. That's the color of scar tissue, often fibrosis of scar tissue. Uh, so when there's laying down of connective tissue because of some pathologic process, it may certainly look grayish like this. So the effect here is somewhat similar to what you see in a cardiac tamponade, instead of fluid forming a layer around the heart, preventing it from expanding diastole, here the connective tissue that covers the heart, because of this constrictive pericarditis, forms a constricting band or layer around the heart, preventing the heart from filling well during diastole, so therefore it cannot pump well during systole. Let's look at the next layer, the myocardium. Here is myocardium. You've seen this before in prior parts of the course because it was discussed when we discussed muscle. It's one of the principal forms of muscle of the body. It is a striated muscle, but it has some, which makes it somewhat similar in appearance to skeletal muscle, but it has some distinctive features which make it different from the striated muscle of skeletal muscle. If we look at this carefully, we have a kind of a giveaway here that is intercalated discs. This is these transverse lines between the end of one cardiac muscle cell and the end of the next cardiac muscle cell. And it so happens that cardiac muscle cells are bound to each other end to end in a chain of muscle fibers. In fact, there's a chain of branching muscle fibers. That's one of the distinctive features of cardiac muscle. It makes it different from skeletal muscle, makes it different from smooth muscle. In any case, the end-to-end point of attachment of cardiac muscle cells to each other is visible as an intercalated disc. Why is an intercalated disc visible? Well, that's a place where you see multiple junctional complexes between the ends of one cardiac muscle cell and the next cardiac muscle cell. These junctional complexes consist of three main types of junctions: fascia adherens, which is an adhering type junction; macula adherens, which is also known as a desmosome; and gap junctions. What these three junctions do is allow the cardiac muscle cells to be coupled mechanically and electronically. The mechanical coupling prevents them from pulling apart during a cardiac muscle contraction. You don't want that to happen. That would be bad for your heart, bad for your health, as you might imagine. And you want them to be electronically coupled so that when the cardiac impulse finally reaches the myocardium, it can be transmitted from one cardiac muscle cell to the next cardiac muscle cell to the next cardiac muscle cell, and so on. Remember, these cardiac muscle cells, although it may not be obvious from looking at a small field, are coupled together in chains of branching muscle cells. That's really what makes up a cardiac muscle fiber, these branching chains of fibers. In actuality, if you study a heart as a whole, the myocardial muscle seems to be arranged in something of a circular or spiral or helical fashion, such that when the cardiac muscle contracts, it tends to contract with a complicated motion, of course, and I'm oversimplifying here, but part of the contraction is as a ringing type action, somewhat similar to wringing out a wet mop. Well, the cardiac muscle wrings itself out, similar to a mop, so pushing the blood out of the heart, squeezing the blood out of the heart in a ringing type fashion. Other features that are important for cardiac muscle: the muscle nuclei tend to be central in their location. You do have to look a little bit to see this well, but for example, here's a cardiac muscle cell with a central nucleus. Here's a cardiac muscle cell with a central nucleus. You have to look for it. It's not always totally obvious, but if you look hard enough, you generally will be able to convince yourself that these cardiac muscle nuclei are centrally located. Another feature that you might see in cardiac muscle is that the fibers are branching. And this, again, is something you need to look for because it's not always super obvious. But let me see if I could find something here. For example, here, this looks like a cardiac muscle fiber, and I think I see two branches here. So this would be a branch point perhaps right here where my arrow is, and then here's one branch coming off, and here's another branch coming off. And, there are some other places where I think I could identify a similar phenomena, but it often is quite subtle, but you could convince yourself if you train your eye to see that. Uh, in any case, in this particular picture, I'm not too worried about it. I think I pretty much know it's cardiac muscle because I see these well-defined intercalated discs and I see central nuclei, so I'm not so worried about identifying branch points, but it is good to be able to see them. Some other things you might see in cardiac muscle is that if you look carefully, often you will see granular material in a perinuclear position. That granular material is often glycogen because cardiac muscle, you would like to have your cardiac muscle have an energy source, even if it's not receiving enough oxygen at the moment. Another thing you might see in cardiac muscle, notably again in a perinuclear position, is lipofuscin. These cells are pretty much end-stage cells. You have them for a lifetime, and they don't, there's no replenishment, no serious replenishment of cardiac muscle cells from stem cells, although people are doing a lot of research in this area. So that because these are long-lived cells, long-lived end-stage cells, when organelles get degraded over time and membranes get degraded over time, over many years, really, some of the material cannot be removed from the cell and it accumulates at the ultrastructural level as residual bodies and at the light microscopic level as a yellow-brown wear and tear age pigment called lipofuscin. And I'm not seeing much lipofuscin in this particular image. I'm seeing more of a pink-like granular material, which I think is more like glycogen. Some cardiac muscle cells, by the way, are binucleated. It is not unusual to see binucleated muscle cells. So don't worry about it. If you see binucleated cardiac muscle cells, that's normal. That's okay. Let's talk about endocardium. Here we see endocardium. It could be rather boring looking in a sense. Uh, Here you see a flattened layer of single layer, I really would say, of epithelial cells, in other words, squamous cells. Right here, this I think is facing an empty space, which really is the lumen of the heart. These are endothelial cells. Endothelial cells. I know these must be laying upon a basal lamina, although I may or may not see the basal lamina easily. They must be laying upon a basal lamina, and that basal lamina must be laying upon a connective tissue layer like this here,
That don't make sense. I just, well, the last.
a sub-epithelial layer of connective tissue. So that's what we're seeing here. Um, Endothelial cells, a basal lamina, sub epithelial connective tissue, and then underneath that is the muscle of the myocardium over here. That's endocardium, the inner layer of the heart, endocardium in its simplest form. This is an endocardium in a little bit more complicated form, perhaps. Here's a sample through the ventricle. Here we see probably a lumen here, which is empty because the blood has been drained out of the heart. And then I presume during life there would be a layer of flattened endothelial cells that is labored EN, I think, for endothelium. Although it is hard to see the endothelium in this image, realize again by handling and by changes that occur after a tissue is removed from the body or the person dies, the epithelial cells can fall off. So that's an artifact. During life, there would be flattened epithelial cells, flattened endothelial cells, and sometimes you see it well in a specimen, and sometimes you don't. In this particular specimen, they are a little bit challenging to see, but it doesn't worry me too much. I know there must be endothelial cells during life. So underneath that is the connective tissue here, the subendothelial connective tissue. And interestingly, in the deeper parts of the endothelium, I see what looks like longitudinally oriented cardiac muscle cells here, muscle fibers, right? These look like cardiac muscle fibers, which are in the deeper part of the endocardial layer. Well, this deep part of the endocardial layer is actually called subendocardium, the subendocardium. So the way it's used in Junkera, and this is the way I will use it because Junkera does it, I will call the deep part of the endocardium the subendocardium. And within the subendocardium of the ventricles, you see a specialized form of cardiac muscle cell which conducts the cardiac impulse more than contracts. Its purpose is to conduct the cardiac impulse, not so much to contract. These cells are called Purkinje fibers. Purkinje fibers. So these specialized conducting muscle fibers in the subendocardial layer of the endocardium are Purkinje fibers, and they look a little bit different from the typical contractile cardiac muscle cells of the myocardium. How do they look different? Well, one thing, they tend to be larger. They tend to be larger than the muscle cells of the myocardium. Although I don't think it's so obvious in this particular picture, but they do, as a whole, tend to be larger in their diameter than the muscle fibers of the myocardium, and they tend to be more pale than the muscle cells of the myocardium. And I think that is true of this picture. They do look more pale than the muscle cells of the myocardium. One reason they look more pale is that they have less contractile filaments within their cytoplasm. They have less myosin, actin, other contractile proteins compared to the typical contractile muscle fibers of the myocardium. And that's one reason they will look more pale. Another reason they look more pale is that they tend to accumulate more glycogen than the contractile cardiac muscle fibers. Now, I know that contractile cardiac muscle fibers do accumulate some glycogen, but these accumulate even more glycogen. They have even more glycogen, as if they need to be able to conduct the cardiac impulse, even if there's low oxygen being provided for aerobic metabolism. They still are able to generate energy to conduct the cardiac impulse. Okay, so here's a nice example of Purkinje fibers. By the way, Purkinje fibers are these conducting cardiac muscle cells in the heart. Purkinje cells or Purkinje neurons are certain flask-shaped neurons found in the cerebellum that we learned about earlier in the course when we studied nervous system. These are actually Purkinje fibers if you're using the words properly. Let's talk a little bit more about the cardiac conduction system. It kind of naturally arises when you talk about endocardium and you visualize the Purkinje fibers, so it brings to mind cardiac conduction system. Here's a diagram showing you a simple version of the cardiac conduction system. This is a complicated subject, which is important in clinical medicine because many people have cardiac arrhythmias. They have a heartbeat which is too rapid, or too slow, or they have an abnormal cardiac rhythm. A normal cardiac rhythm is important for normal cardiac function, so it is a very important subject. But we're not going to talk about all those details. Basically, what you want to know is that the cardiac beat begins somewhere in the what I might call the roof of the atrium. That's not an official anatomic term, but is a convenient informal term to identify where the sinoatrial node is located. The sinoatrial node is a little nodule of pacemaker cells which generate the cardiac impulse, and it's located in the right atrium at its junction with the superior vena cava. That's what you're looking at here. These are actually also modified cardiac muscle cells. They are not neurons or nervous system cells. They're actually modified cardiac muscle cells, and they have some resemblance to cardiac muscle cells, but unlike the Purkinje fibers we just looked at, these cardiac muscle cells are actually smaller and thinner than the usual contractile cardiac muscle cells. They are smaller and thinner, and they're actually more pale looking than the usual cardiac muscle cells. And you saw that in the interactive session this week, I believe, an example of how at low magnification, the SA node seems to stand out because it looks somewhat more pale than the contractile cardiac muscle cells around it. And it seems to be arranged around a special artery going to the sinoatrial node, the sinoatrial node artery, which helps form in a way, a marker for the sinoatrial node. If you find that artery, you're easily able to find the sinoatrial node. But anyway, our main goal here is to realize that the cardiac impulse begins at the sinoatrial node, which is known as the pacemaker of the heart. It so happens that these specialized conducting, if you will, um, in, uh, sinoatrial muscle cells will generate depolarizations on their own without any outside influence. They have a innate ability to generate spontaneous depolarizations. They do it at the highest rate of any cell in the heart. So it so happens that they'll generate cardiac impulses at around 70 beats per minute, which is higher than anywhere else in the heart, and that's why they take over the generation of the cardiac impulse from any other place in the heart. Normally, they will set the pace for the remainder of the cardiac conducting system. So once the SA node generates a depolarization, the depolarization is carried through the right atrium to the floor and the deep part of the right atrium, where you see the atrioventricular node. The atrioventricular node will receive that beat and carry the cardiac impulse forward, but it slows it up a little bit. It doesn't conduct the cardiac impulse quite as fast as it was initially conducted through the atria. And because it slows the cardiac impulse conduction just a little bit, it allows the atria to finish contracting and emptying the atria into the ventricles, allows the emptying of the atria to be finished so that the ventricles have maximally received blood from the atria. And then the cardiac impulse is conducted further. There's a continuous axis from the AV node into the common AV bundle of His, the atrioventricular bundle of His, which travels through the cardiac skeleton. It travels actually into the cardiac skeleton, through the cardiac skeleton, into the upper part of the interventricular septum. The upper part of the interventricular septum is actually not made of cardiac muscle. It's actually fibrous tissue. It's part of the cardiac skeleton itself. It's the so-called membranous skeleton. And then the cardiac impulse is conducted through the membranous skeleton, through the AV bundle of His, to the upper part of the muscular skeleton, where it divides further into the right and left bundles. The right and left bundles here, which travel along the the endocardium of the ventricular septum, and these further branch profusely into the Purkinje fiber network, which then carries the impulse along the subendocardial layer of the endocardium of the ventricles, and then eventually in their distal portion intermingle with the cardiac muscle cells, and the cardiac muscle cells further carry the cardiac impulse throughout the myocardium, because the cardiac muscle cells are connected end to end via gap junctions. So in this way, it's a very simplistic, quick overview of the cardiac conduction system. In this way, the cardiac impulse is transmitted throughout the heart. Here's a picture through ventricular, endocardium, and myocardium. It shows you the endocardial layer. It shows you in the deep part of the endocardial layer, you have these big, plump cells, which are pale and have rather rounded-looking central nuclei. These are Purkinje fibers. They are rather pale, again, because they have lots of glycogen within them, and they have relatively few contractile muscle filaments like the actin and myosin. And in this particular picture, they look a lot different from the myocardial muscle cells found deeper in the heart, in the myocardium, because these are specialized conducting cardiac muscle cells called Purkinje fibers. Here's a higher power view showing you on the left-hand side normal myocardial contractile muscle fibers, and here you see the very big, very pale-looking Purkinje fibers with central, more rounded-looking nuclei compared to those of the contractile muscle fibers. Now, because they contain lots of glycogen, if you wanted to, and usually it's not necessary, but it does demonstrate this phenomena well. If you wanted to demonstrate the glycogen in these fibers and therefore demonstrate the Purkinje fibers easily, you might do a PAF stain, which stains carbohydrates, including glycogen. So here you see magenta staining of the Purkinje fibers, and magenta staining even in a granular pattern like right here, which would be fairly characteristic of glycogen, okay? So the arrowheads are showing you granular glycogen staining, and the arrow just shows you overall magenta staining of Purkinje fibers. Let's talk a bit about the cardiac fiber skeleton. It's convenient to talk about it here because we just talked about the cardiac conduction system, and the cardiac skeleton has an important role in the function of the cardiac conduction system. And also, much of the cardiac skeleton is actually found in the endocardial layer of the heart. Not 100%, but much of it is found in the endocardial layer of the heart. Here we see the cardiac skeleton, and it's a fibrous skeleton. Unlike most of our skeleton, which we think of as being bone and cartilage, this is composed of dense, irregular connective tissue. And when you have a heart and you take sections of it and examine it grossly, you don't really see the cardiac skeleton very easily. But if you know where to look, then you are able to find it. If you have a heart and you remove the atria and you look down at the four valve openings, you see around the valve openings a very thin layer of connective tissue. These are the fibrous rings, which form an annulus in which the four valve leaflets anchor themselves. So the valves anchor themselves onto these rings of fibrous tissue, which are part of the cardiac skeleton. There are some additional structures. I'm not going to talk too much about them. They're, for example, the left fibrous trigone right here and the right fibrous trigone right here, which are also part of the cardiac skeleton. And since we have an image of this here, we could show it. Within the right fibrous trigone is a little opening or canal or channel through which the atrioventricular bundle of His travels from the atria to the ventricles. That's where the bundle of His would be traveling. It has to traverse the cardiac skeleton at this point. Now, one of the main functions of the cardiac skeleton is to electronically isolate the musculature of the atrium from the musculature of the ventricle. That's one of the functions of the cardiac skeleton. And the one place where the impulse can travel from the atria to the ventricles is when the atrioventricular bundle of His travels through the cardiac skeleton right here through the right fibrous trigone. That's one place where travel is allowed. This is important for normal functioning of the cardiac conduction system. You ordinarily want the musculature of the atria to be electronically isolated from the musculature of the ventricles. On the right-hand side, you see an opened heart, and you see someone has dissected out the skeleton of the heart off of the heart, and it would look something like this if you were to dissect away the cardiac skeleton from the heart. And actually, this can be done. Someone who's very expert at cardiac dissection could do something like this. What the cardiac skeleton looks like in this situation, it looks like four fused rings. See, here you see four fused rings, which are the annuli or annuluses to which the cardiac valves are anchored to. And you see the central ring consists of the aortic valve, and then there are several other rings around it, of course. As far as functions of the cardiac skeleton, we mentioned that it electronically isolates the atria from the ventricles. We also mentioned that the cardiac skeleton gives anchorage to the cardiac valves. And the third thing that it does is that it allows the atrial muscle and the ventricular muscle to anchor itself to the cardiac skeleton. This is an anchor point for the musculature of the heart to anchor itself. As you can see, the cardiac skeleton, although it's fairly thin and difficult to see in a heart, it does perform some major functions in terms of um, cardiac structure and function. Here's another section through the heart, and we were talking about how it allows the atrial muscle to be electronically isolated from the ventricular muscle. So right here you see A is the atrial muscle and V is the ventricular muscle. You see how they seem to be isolated from each other by a thin septum of connective tissue right here. There's a thin layer of connective tissue, which is part of the cardiac skeleton, electronically isolating the atrial from the ventricular muscle in this particular picture. This is a very fortuitous picture. It's hard to find something like this, but it demonstrates very nicely the isolation of the atrial muscle from the ventricular muscle. The heart valve, as I said to you earlier, is a elaboration, a projection of the endocardial layer of the heart. And here you see the outer layers of the heart valve consists of mesothelial cells. On the surface of this surface here, there would be mesothelial cells. On this surface here, there would be mesothelial cells, even though they're difficult to see. The inner portion, the core of the heart valve, basically consists of connective tissue. The connective tissue here is continuous with and merges actually with the connective tissue of the annulus, which is part of the cardiac skeleton, and the endothelium, which covers the valve on both atrial and ventricular sides, is continuous with the endothelium covering the remainder of the heart chambers. So essentially, the heart valve, again, is a projection of the endocardium, and its components are continuous with other elements of the heart. The connective tissue in the core is continuous with the cardiac skeleton, and the endothelium is continuous with the endothelium covering the chambers of the heart. Now here we see a chordae tendineae. This is a atrioventricular valve. So there are these tendinous cords or chordae tendineae, which are also composed of connective tissue, which is continuous with the connective tissue of the valve itself, which then will anchor itself to the papillary muscles of the ventricle as well. So the valve is actually a bit of a complicated structure if you really look carefully at it, but it is understandable in terms of its histology, as I just showed you. Let's talk about blood vessels. Let me just check the time here. I want to see what time it is. Okay. The heart consists of 3 layers. The blood vessels consist of 3 layers, at least the arteries and the veins. Capillaries are a little bit different, though they are simpler vessels. Those are basically endothelial tubes. But the arteries and veins clearly have 3 layers. So when you encounter arteries and veins, even if you don't know what you're looking at, if you think it through, you realize you must find 3 layers. What are the 3 layers? The intima, the media, and the adventitia. So you know artery, you must find intima, media, adventitia. Vein, you must find intima, media, adventitia. Let's talk about arteries first. Here's an elastic artery. Here's a drawing of an elastic artery. On the left-hand side, here's an actual elastic stain of an elastic artery. What is an elastic artery? Well, the largest arteries of the body, such as the aorta, is an elastic artery. It's an elastic artery because it has lots of elastic tissue in its wall. It receives a strong beat of blood pressure from the heart because it is close to the heart, and it expands greatly during systole, and it recoils or retracts back during diastole. What this expansion and recoiling does with systole and diastole, it helps maintain the blood pressure at a fairly even keel. There is some variation in blood pressure, but it's not as enormous as it would be if there wasn't much elastic tissue in its wall. You want to you want to be able to prevent the heart from overextending, of course, and you want to prevent the heart from not recoiling, because the blood pressure would fall greatly. If it did not recoil with diastole, the blood pressure would collapse. But by recoiling, it narrows the lumen sufficiently around the blood within the lumen so that the blood pressure does not collapse to close to zero. And this helps smooth out the cardiac, the blood pressure so that it is fairly uniform throughout systole and diastole. So when looking at this vessel, I know there must be an intima, I know there must be a media, and I know there must be an adventitia. The thin layer here is the intima. You see here, it's a very thin layer. This layer on the opposite side is an adventitia. It looks different. Okay, I can make out a thin intima and an adventitia quite readily, and between it is a media with many wavy layers of fibers or lamellae right here. Okay, this middle layer with many layers of fibers or lamellae is is the media in which there are many layers of elastic tissue. The elastic tissue is staining with this particular stain here. Elastic tissue tends to get wavy when it, is fixed or attracts, and that's why it looks wavy here. In actuality, most of these are not so much fibers as lamellae, which are fenestrated. They are fenestrated because they allow movement of oxygen and carbon dioxide and nutrients and waste products through the arterial wall. The arterial wall is fairly thick, and it cannot receive full nourishment from the lumen, so that whatever nourishment is entering into the vessel wall can actually move through the vessel relatively easily because of the fenestrations within the elastic layers. Even that is not enough for a very thick vessel like an aorta. There's additional blood supply coming into the outer layers via the vasa vasorum. There are additional microscopic vessels, microvessels, arterioles, venules, and capillaries in the outer layers of large vessels, such as elastic arteries and even large veins, which helps nourish the outer layers of these large vessels. Here's another picture showing you an elastic artery. The inner layer, which is the thin layer here, is the intima. The outer boundary of the intima is often seen as an internal elastic lamina, a distinct layer of elastic tissue called the internal elastic lamina. The outer layer here looks different. It seems to be composed of more typical collagenous tissue. This is the adventitia, and actually I see little blood vessels out here, a microvasculature, if you will, small arterioles, venules, um, capillaries, which help nourish the outer layers of the wall. That's the vasa vasorum, the vessels of the vessel. But most of the thickness of the vessel wall consists of the media. Here you see many, many lamellae of elastic tissue. They're wavy looking. Now, these lamellae of elastic tissue are made by smooth muscle cells. It's mostly smooth muscle cells where it's actually laid down elastic tissue and ground substance, not so much fibroblasts as smooth muscle cells. So the media here is rich in smooth muscle cells and rich in elastic tissue. Here's another higher magnification showing you an elastic artery. There's a thin inner layer, which is the intima. There's an endothelium lining the intima. Um, this layer here must be the media. It has many, many layers of elastic lamella, which stain with this particular elastic tissue stain. Out here is a little bit of adventitia right here. And this is even further magnification. Here you see an intima. The first elastic tissue layer we encounter at the inner part, just inside the intima here, you might say it's on the inside of the intima, is the internal elastic lamina, and then the remaining layers of elastic tissue would be the lamellae of elastic tissue in the media of this elastic artery here. Here's another picture of an elastic artery showing you even better the adventitia out here. This is adventitia, and within the adventitia, I see a microvasculature, very small arterioles and venules and maybe capillaries, and there may even be some other things like nerve twigs. This is part of the media right here with many layers of elastic tissue. Here's a muscular artery. A muscular artery is not as large as an elastic artery. These are the named arteries of the body, such as the cerebral arteries, the coronary arteries, the hepatic artery, the mesenteric artery, the renal artery. Things like that are muscular arteries. And these arteries don't have all those lamellae of elastic tissue like we saw in the aorta, but they do have lots of smooth muscle cells in their media. If you look at this particular image here, you see a very distinct internal elastic lamina. That's one thing that's fairly characteristic of muscular arteries. The internal elastic lamina is easy to find usually. There are many layers of spindle cells here in the media, which is fairly prominent in these vessels. These are essentially smooth muscle cells. That's why they're called muscular arteries. And here's an adventitia. Now, in the larger muscular arteries, you will also see an external elastic lamina, which I do see here. But usually it's the internal elastic lamina, which is most reliable for identifying and most easy to see in the muscular arteries. And that's what we're seeing here. Here's another picture of a muscular artery. We see a really nice vessel wall. The vessel wall is relatively thick in comparison to the lumen. There's a very well-developed internal elastic lamina here. There's a nicely developed muscular media here with many layers of smooth muscle nuclei, smooth muscle cells seen here. And this is the adventitia here. Now, this happens to be an elastic tissue stain. The elastic tissue stain was done here, but it's actually showing you a muscular artery. So don't be thrown off by the fact that someone is using an elastic tissue stain to show you a muscular artery. It so happens that there's elastic tissue in all the layers of a blood vessel, even, not just the media as we saw in an elastic artery. It can be seen in any layer of an artery. But in the media of a muscular artery, you don't have many layers of elastic tissue, as you can see here. Here's another high power magnification of a muscular artery. I see a nice intima here, and I do see a nice internal elastic lamina here. I see a nice thin media, excuse me, a nice thin intima here, and many layers of smooth muscle here. And here's an adventitia out here. Now, the smallest vessels are that are arteries are arterioles. Those have only one or two layers of smooth muscle. So what's labeled here is an arteriole. A is labeled arteriole. Here I see only one or at most two layers of smooth muscle cells in the wall, and that's it. So that clearly is an arteriole. Now, often arterioles will travel with venules. So in the same picture, although we're not talking about the venous vessels at the moment, we do see a venule next to it, which we will look at again when we talk about the veins. But as you can see, in comparison, the veins are thinner walled vessels and tend to have larger lumina. So since we're seeing this, I'll mention it right now. Veins tend to have larger lumina compared to our arteries because they are the vessels that have most of the blood volume actually. And also their walls tend to be thinner in comparison to the arterial vessels because they have partly because they have less smooth muscle in comparison to those of the arterial vessels of similar caliber. Let's talk about capillaries. There are three main types of capillaries. There are continuous capillaries, fenestrated capillaries, and discontinuous capillaries. Continuous capillaries have endothelial cells, which are fairly, if you might say, solid looking. They don't have many fenestrations or openings within their cytoplasm. The processes or bodies of these cells are bound tightly together by junctional complexes, which contain tight junctions, lots of tight junctions. So junctional complexes containing tight junctions bind together the adjacent endothelial cell processes to help maintain a nice barrier between the blood and the interstitium. And there's a basal lamina around a continuous capillary, which is continuous. The basal lamina is fairly complete or continuous. Now, often on the outside the endothelial cells of capillaries, you do see an externally located cell called a pericyte. So, in essence, capillaries are endothelial cell tubes. They are tubes of endothelial cells, but there will be, to varying extent, cells on the outside called pericytes. We'll talk a little bit more about pericytes in a moment, but here's a pericyte. Here's a fenestrated capillary, which within the cytoplasm of the endothelial cells are many windows or openings or pores, if you want to call it that, which allow more exchange of materials between the blood and the interstitium. But nonetheless, the basal lamina around the entire endothelial tube is still continuous. It's still continuous. This is a somewhat leakier, if you will, a capillary compared to the continuous capillary. Most capillaries in the body are continuous capillaries. So if you look, for example, at skeletal muscle, the capillaries will be continuous. If you look, for example, at brain, where you have a so-called blood-brain barrier, the capillaries will be continuous. They maintain a fairly strong barrier between the blood and the parenchyma of the brain. You need to have exchange between the blood and the parenchyma where there are continuous capillaries, but you want that exchange to be well controlled. In contrast, when you have fenestrated capillaries, you want to have more exchange, more easy exchange, if you will. So, for example, in endocrine glands, where you want hormones to go from the gland parenchyma into the bloodstream, in that situation, you may have fenestrated capillaries. Or in the kidney, where you filter blood to make urine, you may want to have fenestrated capillaries. So there are certain situations where the body needs to have fenestrated capillaries, where you need more exchange to occur, uh, compared to the type of exchange that may occur with continuous capillaries. And then there are the leakiest capillaries of all, which are called discontinuous capillaries, which you see here on the right-hand side here. You have really big fenestrations or openings, if you will, in the cytoplasm of the endothelial cells. You have really large gaps or clefts between the endothelial cells and the least amount of junctions between endothelial cells. You see large gaps between endothelial cells. And the basal lamina itself can be very discontinuous. You see here the basal lamina, it's rather discontinuous, large gaps in basal lamina. This creates a very, very leaky vessel. So another name for discontinuous capillary, by the way, is sinusoid, or sinusoidal capillary. So you see capillaries like this in the liver, in the spleen, in the bone marrow, because those are situations where we would like to have very porous or leaky capillaries. Another feature of these sinusoidal capillaries or discontinuous capillaries is that they tend to be wider than the other types of capillaries. When blood vessels are wider, there's slower flow of blood within them. The flow is slower. So the slowing of blood flow helps maximize the amount of exchange which is occurring between the blood and the parenchyma. Here's a continuous capillary. It looks like an endothelial tube. Here's the body of the endothelial cell, where you have the nucleus, you have cytoplasm, you have the organelles of the capillary. Many of the organelles, by the way, are little vesicles which are transporting materials between the lumen and the interstitium. Because of transport of materials from, even when a capillary is continuous, there still needs to be transport, and much of the transport occurs not so much between the endothelial cell processes, but within the cytoplasm. So one surface to the other surface, between the lumen and the interstitium. And much of this is in the form of transcytosis, movement of materials from the lumen to the opposite side of the cell. That process is called transcytosis, and much of that may occur by movement of these vesicles from one side of the cell to the other side of the cell. Outside these endothelial cells, I see a rather pale gray line. That pale gray line is continuous. That is the basal lamina. In this particular picture, I don't see any pericytes, any outer cell on the outside of the endothelial cell. Here I see an endothelial tube, which is the continuous capillary, but on the outside I see another cell embracing the capillary. You see the cell here embraces the outside of the capillary. It sends processes, in other words, embracing the outside of the capillary. That's a pericyte. Now, what do pericytes do? Pericytes, they have contractile filaments within their cytoplasms. They may be contractile, so they may help control the flow of blood within capillaries. They are not smooth muscle cells. They are pericytes, but they do have the capacity perhaps to control the caliber of the capillary and move and flow of blood through the capillary. They also may act as stem cells, mesenchymal type stem cells, and even in adults. So they may be able to, if there was injury or need to make new capillaries, they may be able to give rise to endothelial cells, smooth muscle cells. They may be able to give rise to non-vascular cells, even non-vascular mesenchymal cells, such as bone cells, osteocytes, and cartilage cells, chondrocytes, and even other mesenchymal cells as well. So they do some important things. Now, pericytes also have a basal lamina, and here the basal lamina is labeled basement membrane, BM. So I said to you, endothelial cells have basal lamina. Well, pericytes also have basal lamina, and the basal lamina of pericytes and basal lamina of endothelial cells merge or are fused together if you look carefully. Another thing you see in this picture is that at the junctions between endothelial cells here, you see some dark staining. This dark staining are the junctional complexes, including adherens type junctions between endothelial cells. So that dark staining reflects these junctional complex. There's also a little bit of a protrusion of endothelial cytoplasm into the lumen here. That's a so-called M marginal fold. So I see some marginal folds right here where there are cell junctions between two separate cell processes. Here we see basal lamina. In this particular case, they're labeled BM, basal membrane, but here you see a basal membrane or basal lamina of an endothelial cell on that side, and here you see a basal lamina, basal membrane of a pericyte right there, of a pericyte. P is for pericyte. Here we see even higher magnification showing you the two cell processes coming together and being joined by cell junctions right here. The dark staining are cell junctions, and here we see a marginal fold right here protruding into the lumen. This is a marginal fold in a capillary. It is not a valve. It is not a valve. It is a marginal fold. There's a difference. Capillaries do not have valves. They do not have valves. They have marginal folds. Okay? Capillaries have marginal folds. They do not have valves. Here we see a scanning electron micrograph showing you, it's very Halloween looking to me, kind of creepy crawly looking. These pericytes on the outside of the capillary tube, they send these delicate processes surrounding the capillary. Here we see again pericytes on the outside of the capillary tube again. Just from the location, I could tell what's a pericyte and I could tell what's an endothelial cell. Here's a fenestrated capillary. I recognize it as a fenestrated capillary, partly because I see lots of arrows, but even without the arrows, I could see there are fenestrations here within the capillary endothelial cytoplasm, and I see actually the fenestrations are bridged by little diaphragms. There's a little line across the fenestration, which are diaphragms across the fenestration. They are not basal lamina. A fenestration is bridged by a diaphragm, not by a basal lamina. These endothelial cells do have basal lamina. That's this arrow right here on the outside, the gray line on the outside. It's a double arrow, actually. The double arrow on the outside is pointing towards the basal lamina. This arrow is pointing towards a diaphragm across a fenestration. This is a very leaky looking capillary. Actually, it's a very nice picture. It's also called a sinusoid. It's a fairly wide capillary. These happen to be hepatocytes. This is liver. These are hepatocytes. The surface of hepatocytes facing the capillary has lots of microvilli. Why does it have lots of microvilli? To increase the surface area of the hepatocytes facing the capillary. The liver is the metabolic core of the body, if you will, the metabolic center of the body. That's many, many very important things. So it's having all these microvilli maximizes the exchange occurring between the sinusoidal capillary and the hepatocytes. Well, these sinusoidal capillaries are very porous looking. They're huge fenestrations within the cytoplasm of these capillaries, number one, there are large gaps between endothelial cell processes, right? Large spaces between one endothelial process and the next, large gaps between endothelial cells. And number three, there's large discontinuities in the basal lamina of these endothelial cells. It's hard for me to even find basal lamina here, which is really strange. Ordinarily, you expect to see basal lamina quite easily around an endothelial cell, but here you can't find them. It's almost as if there's almost free exchange between the lumen of these sinusoidal capillaries and the hepatocytes. It's as if the metabolic functions of these hepatocytes is so important that it needs to have very porous capillaries to do its job. Let's talk about veins. Let's start at the smallest veins here. We start at the largest arteries. We went down the artery, the smaller and smaller arteries to the capillaries. Now we're returning to the heart by starting with the smallest order of veins. We already saw this picture. We saw what an arteriole looks like. An arteriole has one or two or maybe three layers of smooth muscle cells in its wall. That is the media of the arteriole. It's even hard to make it out of the media. It's so thin. I see, by the way, some endothelial cells here in the lumen of the arteriole. To the left of it, I see a venule. A venule is larger in diameter compared to an arteriole. In other words, the vein structure is larger in diameter compared to the arteriole because the lumen is larger in its volume compared to the lumen of an arterial vessel, because most of the blood of the body is actually in the veins. In the veins, not the arteries. There's more blood in the veins than the arteries. That's how I remember that. The wall of the vein structure here, in this case a venule, is thinner than that of the comparable arterial vessel. The wall of this vein is thinner compared to a comparable arterial vessel, the arteriole here. And because the wall of the vein is thinner and there's less smooth muscle in the wall of the vein, veins in general tend to be more maybe softer and more collapsible. They tend to flatten out a bit. So the vein lumen and vein overall looks a little flatter, a little bit more flatter, perhaps compared to the arteriole even. We'll see this phenomena repeated as we get into larger and larger venous vessels. Here we have a medium-sized vein. I know there are going to be three layers in a vein. So if we look at this carefully, I know a thin inner layer is going to be the intima. So this thin layer here, it looks visually or morphologically a little distinct. Even if I don't quite understand what I'm looking at, I know that something here, this inner layer, looks a little distinct. This must be the intima. It is lined by an endothelium and has a subendothelial connective tissue. I see another layer right here, like right here. See right here. This must be the media. There must be some smooth muscle in the media, but the media veins tends to have less smooth muscle, less organized smooth muscle compared to that of the comparable arterial vessels. Okay, but I know there must be some smooth muscle cells in here. Whether I can recognize them easily or not, I know there must be some smooth muscle. It's just not as obviously muscular or well organized compared to an arterial vessel. And then I know the outer layer of the vein must be adventitia. So here I see adventitia. Now, one thing about veins, the thickest layer in veins tends to be the adventitia. The thickest layer in arterial vessels tends to be media. So in this particular vessel, the thickest layer is the adventitia. If it in the comparable artery, the thickest layer would have been the media. Here's a large vein. What distinguishes a large vein? This I find a little odd, but it helps me identify this as a large vein. For example, a vena cava. A vena cava is a large vein. Well, there's going to be a thin intima, okay? So this thin layer right here, this is the intima. This layer here, it's another layer. This is going to be the media. It's quite thin. I know the thickest layer of all is going to be the adventitia. This thick layer here is the adventitia. Now, what's strange about the adventitia of a large vein like the vena cava is lots of longitudinally oriented bundles of smooth muscle. You see here the longitudinally oriented bundles of smooth muscle in the adventitia. That's typical for a large vein. You don't see that in the arteries. You'll only see that in a larger vein, such as a vena cava. Now, when you cut the longitudinally oriented bundles of smooth muscle in the vena cava or other large veins, they look like this. These round things here are rounded, longitudinally cut bundles of smooth muscle. That's what we're looking at here in a large vein. That's distinctive for a large vein. Okay. Now, when you actually look at tissue in a body, often arteries, veins, and nerves travel together in neuromuscular bundles. And usually the arteries and veins and even the nerve are of a similar uh, uh, order of size, similar order of size, okay? So here in this particular tissue, we see a muscular artery and a medium-sized vein next to each other, and we see even a nerve of comparable size. So the muscular artery, how do I recognize it as muscular artery? Well, um, the thickest layer I could see quite easily is the media. I see a really well-developed, nice, easy to find internal elastic lamina. Even at this magnification, I think I see multiple layers of smooth muscle cells that are cells with spindly nuclei encircling the vessel, right? Encircling the vessel. The whole vessel has kind of a rigid, rounded, rather rounded appearance, not entirely, but it's kind of oval to rounded, okay? Even in this particular case, although it's not as reliable, I see an external elastic lamina, and here's an adventitia, but probably the best developed layer in this vessel, I would argue, is the media, which is muscular, okay? Next door to it, I see a vessel with a larger diameter, a larger lumen with a greater volume, which is what I would expect to see in a vein. I don't make out an easy to identify internal elastic lamina. I don't make out a well-developed muscular media. I think there is a media with smooth muscle cells in it, but I don't think it's as well-developed or easy to see as you see in the adjacent arterial vessel. And arguably the thickest layer of all in this um, medium-sized vein is the adventitia. Here's the adventitia right here. It looks like it's the thickest layer of all and thicker than the media. Okay, so those are all features of a vein. Also, because the media in the vein is thinner and less muscular, and the lumen is bigger and contains more volume, the vein more easily collapses, gets flattened or irregular in its contour. So just the overall contour of the vein and overall size of the vein help speak for this being a venous vessel. And here's a nerve. How do I know it's a nerve and not a blood vessel? Well, it's a solid cylinder. I know inside are going to be axons and myelin sheaths and lots of Schwann cells. The nuclei here are mostly Schwann cells. So this solid cylinder is a nerve of comparable size. What can go wrong with vessels? There are lots of things that go wrong. If you're talking about veins, veins have valves. So the veins in the legs, if someone stands a lot, or if from aging, or from having multiple pregnancies or whatever, which put more pressure on the veins, causes some loss of competence of those veins. Um, the blood may start pooling in the leg veins, causing dilatation and tortuosity in the leg veins, and you get what we may recognize as varicose veins. Easy to recognize varicose veins. Here's a cross section of a vein like you might find in the leg, and you see valves. So veins have valves. Veins are a low pressure system. And in order for you want, and the veins and legs, in addition to being low pressure, also have a tendency to refill with blood because of gravity. So to encourage a unidirectional flow from the legs back to the heart, have valves, especially the veins in the legs. The valves prevent backflow of blood into the dependent portions of legs. If you have increased backflow of blood in the veins of the leg, it'll force fluid out into the interstitium, and people will develop edema of their legs. And also, people have poor blood flow, who have poor blood flow in the leg will develop changes from ischemia, low blood flow, to the leg. So they get things like stasis dermatitis, ulcerations, even serious infections of the legs, or serious wounds of the legs which don't heal. So this could be much more than just a cosmetic problem. So important to realize that veins have valves. Our arteries don't have valves, capillaries don't have valves, veins have valves. And that's what we're seeing here. The valve of a vein is also an elaboration of the intima of a vein, just like the heart, the inner layer of the heart, the endocardium is where the heart valves come from. Well, the valves of veins come from the intima, the inner layer of the veins. Here we see a little bit more about varicose veins. Let's talk about lymphatics. The last blood vessel we're concerned about lymphatics. Now, there are two circulatory systems in the body, actually. There's the blood circulatory system, which we've been talking about, but when blood flows to tissues and fluid exits into the interstitium of the tissues, and the fluid actually is flowing through the interstitium, it's not stagnant or in the interstitium, it actually is flowing in the interstitium. Most of the fluid returns back into the blood vasculature to be returned to the heart, but some of the fluid, does not return back to the heart. So that fluid must be picked up. The excess fluid, which does not return to the heart at the venous end of capillary beds, is returned back to the blood vascular system via the lymphatic system. How do we recognize the lymphatic in a tissue section? Well, here's a tissue section. I'm not sure from where. Here's a gland of some sort. Here's a small arterial vessel, which is an arteriole. Here's a small a venous vessel, which is a venial. Notice the arterial has a smaller diameter, a smaller lumen, and a thicker wall. Notice the venial has a larger diameter, a larger lumen because it stores more blood, and a thinner wall compared to the arterial. But that's a side issue in this particular picture. The thing that I want to show you is that the lymphatic vessel has the thinnest wall of all. It's very, very thin. It's very, very thin here compared to the arterial vessel and the venous vessel. Very, very thin. Also, within the lumen of the lymphatic vessel, there are no red blood cells. There are red blood cells in the arteriole. There are red blood cells in the venial, but no red blood cells within the venial. You may see some lymphocytes, even some white blood cells within a lymphatic, but you will not ordinarily see red blood cells. So here's a lymphatic vessel to round out the blood vessels of tissue. Lymphatic capillaries have a special structure which helps them in their function. Lymphatic capillaries is where the flow starts within the lymphatic system. Lymphatic capillaries are blind-ended. They just end blindly in tissue, and there are openings between one endothelial cell of the lymphatic capillary and the next endothelial cell of a lymphatic capillary, and so on. So excess interstitial fluid because of the hydrostatic pressure within the interstitial tissue forces itself into the lumen of the lymphatic capillaries, and therefore it drains into larger and larger lymphatic vessels, and eventually is returned via the largest lymphatic vessels to the blood vascular system, that is via the right lymphatic duct and the thoracic duct in the two veins of the neck area. Now, because these are very thin-walled vessels, and you want to keep them open so they could receive excess tissue fluid. The endothelial cells of lymphatic capillaries are anchored to the interstitial connective tissue via anchoring filaments. So here we see anchoring filaments anchoring these lymphatic capillary endothelial cells to the interstitium. There's some elastic tissue in those anchoring filaments and collagen in those anchoring filaments. When the fluid flows into the lymphatic capillary, actually the endothelial cell forms little flaps that close off these openings so that the fluid is flowing unidirectionally into the lymphatic capillaries and not returning back, not regurgitating back into the interstitium. It's important that the flow into the lymphatic system be unidirectional. Now, because of the low pressure in the venous system, much of the force that pushes the venous blood back to the heart is from motion of the body. So when the body moves or when the muscles contract, that helps move the venous blood back towards the heart, and the flow in the veins tends to be unidirectional because of the valves within the venous system. Well, something similar occurs also. By the way, there is some smooth muscle in the wall of venous vessels as well that could also be involved in motion of of the of the blood going towards the heart. Now, in terms of lymphatic vessels, there may be a little bit of smooth muscle in the wall of these vessels, but much of the contractile force for movement of lymphatic fluid back towards the venous system and back towards the heart comes from contraction of muscles and movement of tissues, by exercise and various movements of the body. But again, in addition to contraction of muscles and motion from various activities, you want to encourage the flow of lymphatic fluid to be unidirectional back towards the heart, and that's also done by the fact that lymphatic vessels can also contain valves. So here we see a lymphatic vessel. This is not a lymphatic capillary. This is a little bit of a larger vessel, but here you see a valve within a lymphatic vessel encouraging unidirectional flow of lymphatic fluid back towards the, a larger lymphatic vessel, and eventually returning to the blood system and the heart. The lymphatic valve, again, like the other valves we talked about today, are elaborations of the inner lining of lymphatic vessels with an endothelium covering the valve and a little bit of connective tissue inside the valve leaflet, like that. So these are lymphatic valves found within lymphatic vessels. And with that, I have finished my talk today. Is there any questions that you want me to answer before I stop the recording?